This is the component 1 task book, and we are up to question 5, which is microbiology. The graph shows growth of 2 colonies of the same bacterium. And they're using 2 different carbon sources, one with glucose and one with lactose. Immediately you should recognize, hang on, I know about these. Glucose is a monosaccharide. I could draw glucose. Lactose, I know that's a disaccharide made out of glucose and galactose. So you already know some information about that. Estimate the rates of growth for the 2 colonies between 80 and 100. Well, we can actually calculate it from looking at the graph. So our rate is the growth divided by the time. So our growth is the number of cells over the time. So what we're going to do is, for glucose, we go, what's the time? Well, the time is the same for both of these, so it goes from 80 to 100. And actually the points are on here as well. So it's these points here. And we're going, that's our time taken. And so our time taken. And our growth is how far it's gone up. And so our time taken is actually 80 to 100 minutes, that's 20 minutes. Our growth, well, with glucose it's gone from 22 to 25, so it's gone up by 3, so 22 to 25 is 3. With lactose it's gone from 16 to halfway, so 17 and a half. It's gone from 16 to 17.5, so that's gone up by 1.5. So our first rate calculation will be for glucose. So glucose has gone up by 3 growth in 20 minutes. That's going to be our calculation. So 3 divided by 20. Our lactose is going to be 1.5, again divided by 20. Trusty calculator, 3 divided by 20 is 0.15. And the lactose one, 1.5 divided by 20 is 0.075. We need some units. Numbers are meaningless without units. So our growth rate is our growth, there, number of cells times 10 to the 4 millimetre minus 3 per minute. So that's going to be our growth. So cells times by 10 to the 4. Should be 10 to the 4 millimetre minus 3 per minute minus 1. So that'd be the full unit. So I'm just using that unit there, that description divided by time. If you said cells per minute, well, it's not true, is it? Cells, it's not 0.15 cells, it's cells to the power of 4, and it's per unit volume. So it doesn't make sense without the rest of it. You might have expressed it slightly differently, per 20 minutes or whatever. That's fine if you do an alternative version of this, but we've got a number and a unit. Suggest a difference, an explanation for the difference between the two colonies. So we're saying, why does that one, glucose, grow faster than that one, lactose? Well, you've done respiration. So you know that respiration starts with glucose, and if we're going to use lactose, well, we're going to have to hydrolyze that bond to get our monosaccharides. We might even have to convert our galactose into glucose, and that's going to mean a delay in the growth. So that's the kind of favourite explanation. There are some others. It might not use lactose at all. Lactose might not be absorbed, but that's a nice biological argument that I'm happiest with. So, enzyme needed to hydrolyze or convert lactose to glucose, or possibly glucose is the starting point for respiration. So something along those lines. Part C, apart from the carbon source, and some people have put the carbon source in, name two other nutrients likely to be included. Well, the section in your notes here, what do microbes need for growth? They need a carbon source to give them energy, and then they're going to make their own protein, so they would need a nitrogen source. But we need to be careful with what we're saying here. So we need a nitrogen source, but we need to say what sort of thing would be included in the medium. You can't add nitrogen to the medium. Nitrogen gas that just bubble through and won't be able to be used. You might not have done nitrogen cycle yet, but you can't use nitrogen directly. You'd need some other form of it. So for example, nitrate, amino acids, ammonium ions, any sort of reasonable suggestion here. So I'm going to put nitrate. And other things that you need, if you've done nutrient cycles, you might mention phosphate, but certainly something that's definitely in your notes, you need kind of vitamins. I can't spell it. Vitamins and minerals. Some people have put vitamins and minerals as the two separate points, but they're not different enough. They wanted, the major groups that you should have known. The results were obtained using the viable counting technique. Explain briefly how this is carried out. So this was, you've got your original culture with the bugs in, and then you did a serial dilution. You did this with milk, and this had, for example, 9 of sterile medium, and you added 1 cm cubed, mixed it, 1 cm cubed, mix it, 1 cm cubed, mixed it, and then took volume, for example, 0.5, it could have been anything there, and put that into an agar plate, and then you counted the colonies, and then did some maths. So that's what they wanted. They just wanted a brief overview of that. So for example, use sterile conditions, unless it won't work because it'd just be full of contaminants, or aseptic technique, another word for that. And I'm going to mention serial dilution. Serial dilution, and you could give an explanation of that. So, of a known volumes. That's our known volume there. So 1 cm cubed, for example. What are we diluting it in? Into sterile medium. But you could say, sterile water, saline buffer. Just an idea of it's not got the bacteria in. And then, for example, 1 to 9 cm cubed, that sort of thing. And then finally, so you got your 1 to 9 described, and we're saying how much we're putting onto our plate. So known volume, e.g. 0.5 centimeter cubed, added to agar plate, and you could have done a pour plate or a lawn plate. Incubate at 25 degrees C for 24 hours plus. Not using 36 because that would encourage the growth of pathogenic mammalian bacteria, which we don't want. And we're going to count the colonies, then calculate the original density of bacteria, allowing for the dilution factor. So you just needed 4 of those, and most people got 3 at least with that one. Why is this procedure called viable counting? Well, because the cells need to be viable in order to make a colony to grow. Any dead cells won't grow, so it doesn't include dead cells. So this ignores dead cells, counts only living cells, able to reproduce. And finally, just to make a paper add up somewhere, they've added another question. Why would they use something else like a colorimeter instead? So they wouldn't do this time-consuming and expensive procedure. They just use a colorimeter. What's a colorimeter do? A colorimeter, well, let's stick with our original diagram. A colorimeter would shine some light through there, and some would get through, and they would have a detector here and see how much light gets through, and more bacteria, less light would get through. But the advantage of that, quick, easy, cheap, certainly cheaper than this, and that's what we want, and that's what industry wants. So less cost, for example, quicker, less labour intensive. The downside is that you're going to count some dead cells, but in industry it does take that into account. So a nice question for microbiology.